Okay, so we're gonna go ahead and get started. We're kind of all over in the Science Learning Center, so I'm gonna be like yelling a little bit. But if you can't hear me, just let me know and I can talk louder, believe me. So we are going to be going over what you need to know for your first lab practical for Anatomy 1. Some of you will begin taking it today at like 11:30. So I'm going to try to go through some of the things we learned in the first couple of weeks kind of quickly so we can spend some time with tissues, because I know that is usually what a lot of the students have trouble with. So these are the exercises that are going to be covered on the lab exam. It's 1 through 6, and these are just all of your worksheets that you should be completing and then turning in. A lot of the same pictures from those assignments are going to show up on the lab exam. So if you have not completed those, definitely use those as a study tool. So it should be like language of anatomy. Those are the directional terms, the regions, the microscope, a little bit of chemistry. It's not as in-depth as we do in lecture, but it's the pH scale and something else I already forgot. The cell, anatomy and division, cell transport, and then the big one is the tissues. Oh, wrong one. Okay, so the language of anatomy, you're going to want to be able to describe the anatomical position in your own words. You always want to kind of get it out in as few words as possible, but make sure you explain yourself really well. So you don't need to write 2 or 3 sentences, right? One sentence will be enough as long as it's a quality sentence. So can anyone in a few words describe the anatomical position? Flat feet, palms out, arms to the side, thumbs pointing where? Lateral, good. See, so that's a good example of using anatomical terminology to answer the questions, thumbs laterally. Another thing on your exam will be the body planes. So median or mid-sagittal. The thing with this one is it can always be sagittal. Mid-sagittal means it's directly in half. So I can cut a bone into left and right parts, but unless I cut it directly down the middle, it's not mid-sagittal. So if I cut a bone, I wish I had an example of something. If I were to cut this torso up here, right on this line right here, that would still be sagittal because I'm cutting it into left and right parts, but it's not mid-sagittal because it's not right down the middle. So that's something I think that students get confused sometimes. So again, mid-sagittal, this would be an example of that. Frontal or coronal, and then transverse. So the way that this can be asked on the exam is a cut of the transverse planes separates the body into blank and blank parts. What would the answer be? Say again. Upper and lower, but superior and inferior. Superior and inferior, same thing with frontal. It cuts the body into what parts? Ventral and dorsal, or anterior and posterior. So you want to make sure you're able to answer questions like that. And then again, there's a specific image of the lady in like a tank top and leggings that's going to come up on the exam. So you want to make sure you're familiar with that. That image should also be in the Kahoot if you have access to that already. And then I'm going to skip forward and backwards a few times. So body landmarks are these right here. You're going to want to make sure you're comfortable with those. We do have extra of these printed out in the lab that you can use while you're in here. We can't let you leave with them, but these are available to you in the anatomy Canvas tutoring shell. If you need help with that, let me know. But you should be familiar with these. Most of these are going to be just straight up labeling. So for this guy, he does show up on the exam. I want to say this anterior portion is a whole station, and then the posterior portion is going to be its own station. So you can expect at least 8 blanks for this guy right here. And then body cavities are these over here. The way this can be asked is just to name the body cavities, or for example, I don't know if you guys can see this far, but number 2 up here, what cavity is this? Cranial cavity. So a question they can ask, identify the cavity, cranial cavity, identify an organ or structure inside of this cavity. What would that be? Brain, good. And they can do that for any of the cavities. One thing I am seeing a lot of with the students is they're getting the ventral cavity and vertebral cavity confused. The ventral cavity is going to be anterior, and the vertebral cavity is going to be posterior. The vertebral cavity holds your spinal cord and vertebrae. Vertebral cavity, vertebrae, and that makes up your spine. So just remember that. Ventral is referring to the anterior cavity. So don't get those two confused. What is the structure that separates the, abdominal pelvic from the thoracic cavity? Diaphragm, good. You're also going to want to know that. That will most likely come up. And then just being able, like I said, to label the cavities and then knowing what's going on inside of them. And then lastly, we have our quadrants and regions. So let's start with this guy and let's name them. What is this one right here where my pointer is at? Left upper quadrant, let's move down. Left lower, and then over here. Right upper, and this one. Right lower quadrant, good. So when you're doing this on your exam, what I suggest for both the quadrants and regions is when you finish a question, and it could be any question that you are pretty confident on, that you know exactly what the answer is, draw a little tic-tac-toe, what do you call it, like board on your exam, and then start filling in the quadrants or regions because you know that question is going to come up. So when you get to that specific question, you don't have to really think about it, and you already know that's there. And then let's go over here. What is this region? Or I'm sorry, yeah, this region. Left hypochondriac. So this one up here is going to be the left hypochondriac. Does anybody know what chondra means? Did you say cartilage? Yeah, cartilage. So going kind of jumping into chapter, was it 6 or 4? Cartilage, this is hyaline cartilage. It connects your ribs to your sternum. So chondra means cartilage, left hypochondriac. This one, left lumbar, because of your lumbar vertebrae. And then this one down here, iliac. Make sure you, on your exam, you are specifying left and right. So we know through all of these. So we already know these. Let's go to the middle, epigastric and hypogastric, or pubic region. I believe whatever it says in the book should be correct. So if it says hypogastric and then pubic in parentheses, that would also be correct. So epi, umbilical, hypo, I don't think I said that for the Zoom people. And then we already named these over here, but let's go through them. What is this one? Right hypochondriac, lumbar, and right iliac, good. And then you're going to want to know at least one organ from each of these. So let's go through that quickly. Just can anyone name one in the left upper? Stomach, small intestine. This one can be gallbladder, liver. And then this one down here, large intestine, yeah. The bladder could also go in either of these. Over here, it gets a little trickier just because they're cut up into kind of smaller regions, but same thing applies. You would need to know an organ in each of these. I know it's pretty tempting to write like ribs right here, but unless they ask or read the question, they might ask for a specific organ and not just a structure that's here. Lungs. I don't think that would qualify as being in here because the lungs are going to be more up here. in the thoracic cavity, and this is more the abdominal pelvic. Okay, and then I believe that's it for chapter 1 or exercise 1. Now we're going to move into the microscope. You're going to want to be able to identify all components of the microscope. So all of these that are numbered here. In the lab, we do have some microscopes that are already like just set out if you want to work with them. You should expect 2 to 3 questions directly on microscopes, and this can be like just answering like anatomy of the microscope, specific parts, or like short answer questions. So for an example of a short answer question, as the magnification increases, the space between the objective and slide will decrease. This space is called the working distance. So this is an example of a short answer question that you can have regarding microscopes. Another one, what must be used within the 100x objective to minimize light refraction and improve resolution? I heard someone say oil. What type of oil? Immersion oil. Good. And then another one, how do you calculate total magnification? We use 2 specific parts of the microscope. What was it? Not the fine knob. So we use this up here, the ocular lens and the objective lens. What is the magic number when calculating total magnification? 10. So it's always going to be, what is it? Objective lens times 10. So your answer should either be, let me do math, 40, 100, 400, or 1,000. If you're putting like 4,000 or 10,000, not the correct answer for our microscopes that we have in the lab. And you're going to want to make sure that you can identify which objective lens you're looking at without looking at the number, because I do believe they cover the number. So I'm not entirely sure if they cover the color. They might, but I'm not entirely sure if they do. But the way to tell is I always tell students to start or find the smallest objective lens first, which would be the 4, and then move your way towards right over the light. So right here on the stage to see which one is pointed inside of that area or pointing to the slide, and then count your way towards it starting at the 4. So if you start at the 4, then you know the second smallest is going to be 10, and then 40 and 100. The largest one for our purposes is always going to be 100. And then another way that some of the questions are worded are like name the function of x. So they can ask you, let's say number 1, identify what number 1 is. What is number 1? Ocular lens, and then they can say name the function of number 1. And so you would have to name the function to see the slide. And then another short answer question, as the microscope magnification increases, what happens to the size of the field? It decreases. On your exam, if you were to just write decreases, that would warrant full credit. Remember, you only have, I want to say it's 2 minutes per station, so you don't want to be there writing a full-on novel. You really want to get your answer out and then move on to the next question. Any questions about microscopes? The big thing is going to be being able to identify the parts of the microscope and then total magnification. Total magnification will 100% be a question. So it should be, again, your answer should be 40, 100, 400, or 1,000. Anything other than that, probably wrong. No. Okay, and then metric units, you're going to want to know the basic metric units of measurement for the following: length, mass, volume, and temperature. Length is going to be meter, mass is going to be gram, volume is going to be liter, and then temperature is going to be Celsius. temperature is the one, or I should say Celsius is the one that they're going to make you write out and spell out completely. So make sure you know how to spell Celsius and not just write C. And then I know the metric system, some of the handouts that we have are pretty daunting. They're pretty scary. You don't, let me be careful with my words. You should know about every single one, but the ones that they usually ask about are micro, milli, and centi, and then kilo as well. So again, micro, milli, centi, and then kilo. Now, I'm not saying you don't need to know the other ones, but if you're going to spend a lot of time with the metric system, spend some time with those four. These down here, these are positive and then negative up here. I know it's hard to see. Additionally, you will need to know the factors for some of them. So like I said, micro, milli, centi, 10 to the -6, 10 to the -3, and then 10 to the -2, and then same for kilo. Yeah. On the practice exam that's in the Science Learning Center, I do have a couple questions on metric units, so that can help you with that. Additionally, I want to say it may have come up in your lab quizzes. What else was from here? I think this is basically it that you need to know for the metric system. Okay, now with chemistry for your lab exam, it really focuses on mixtures and the pH scale. Mixtures are a little weird. There is a video on Canvas that you can watch that I highly suggest you watch because some of the lab questions come from that video. And the specific video is a professor going through the different types of mixtures in her hand. And she's telling you what the mixtures are. So I think, for example, one of them is like mayonnaise, and then I think it's Kool-Aid, an Italian dressing. And then some of the questions on your lab exam are like, what type of mixture is mayonnaise? So definitely watch those videos. If you don't have access to them, come see me and I can share it with you, or try to, but it should be on your Canvas page. So when we cover mixtures, we're talking about homogeneous mixtures and heterogeneous mixtures. Homogeneous are solutes that are equally distributed. So you can see here it has like a nice little pattern and it looks really nice and pretty. That is going to be a homogeneous solution or mixture, sorry. And that is also down here when we have sugar and water, and that's sugar water. It's equally distributed and it creates sugar water instead of just having sugar plus water. So it creates that mixture that's equally distributed. Heterogeneous mixtures are solutes that are unequally distributed. And so in this example, we have sand and water, and we can see that the sand settles out at the bottom. And this is going to be heterogeneous. I do have a bit more about these on the next slide, but just so you guys can see that example, this really helped me in understanding what the big difference between the two are, is equally distributed versus unequally. Okay, so sorry if that's a little bit hard to see, but we have three types of mixtures: solutions, colloids, and suspensions. Does anybody know what the other word for colloids are? Emulsions. Sol-gel is a type. So with solutions, these are homogeneous, which again, that would mean it is equally distributed. And then solutions also have solvents and solutes. So solvent is the substance in the greatest amount. Does anybody know what the universal solvent is? Water. Good. That's probably going to be a question, so make sure you know that. And then a solute is the substance in its smallest amount, or smaller amount, excuse me. And then we have colloids, which are also called emulsions. These are heterogeneous mixtures. One like characteristic of a colloid is that they're translucent or milky. I believe that will also be a question, like describe a characteristic of, or I have it right here. Include a specific criteria property for each of the following mixtures. So that could be like translucent or milky, or you could just write heterogeneous. An example of a colloid or emulsion is cytosol inside of the cell. That's that like liquid watery substance. And then suspensions, these are heterogeneous mixtures with large solutes that settle out. So like in our example, that was the sand and the water. In the body, an example of a suspension is blood. Your plasma will settle out, and there's a good image of this in your textbook. And then an example of the way this will show up on your lab exam. Include a specific criteria property for each of the following mixtures. A solution like bottled water. What is a specific criteria property? What? A larger amount, is that what you said? Larger amount. You could say larger amounts. Another like one word answer, homogeneous. And then suspension, what is a criteria property? Heterogeneous, yeah. So that would probably be like the easiest answer to give, just straight up describing it. And then another question that can come up on the exam, provide an example of the type of mixture that can be found in the human body. So like I said, with plasma, with suspensions, if they ask you to identify a criteria for a suspension, and then afterwards, so we'll say like part B, provide an example of the type of mixture that can be found in the human body. What do we say? Plasma or blood. I think it'll tell you. Like it should tell you like provide an example of a solution in the human body or something like that. But yes, make sure you watch the video on Canvas for mixtures. It's very helpful. I want to say there's 2, but I'm not entirely sure. And then the second part of chemistry that we cover in the lab is the pH scale, which is right here. pH scale runs from blank to blank. What are the missing numbers? 0 to 14, very good. This will be a question. Don't let it trick you. 0 to 14. The pH scale measures how acidic or basic a substance is. So right, the increasingly higher we go, the more basic it is. Lower, the more acidic it is. The way this will be worded, use the scale provided to answer the following questions, and then it'll ask you a couple questions. What is the term given to a mixture that has a pH given of 7? Neutral, good. So you want to make sure you know that 7 is this neutral zone right here. And an example of something neutral in our body is blood. Yeah, very good. So there isn't a ton of chemistry questions on the lab practical. The lecture kind of covers most of it, but it's more putting these into application in the human body. Okay, and then we get into chapter 3, the cell anatomy and division. So I have 2 images up here of the cells. You're going to want to be able to label both the like cartoon picture and this animal cell. We also have it back here if you need to practice with it, because both of these will show up on the lab exam. You'll need to know the anatomy of the cell and the function for each of the organelles. I'm going to kind of run through a few of them quickly with you, but if you came to the lecture review, we already went over this. So all of these inside of the cell, do we know what they're called? Organelles, yeah. So they're like little organs that keep the cell running. First, we have the mitochondria. Does anybody know the function? So Powerhouse, yeah, so it's the site of cellular energy production, right? So mitochondria, energy. We have our ribosomes. These are the site of protein synthesis. We have our endoplasmic reticulum, and we have two types. I don't think you guys will be able to see, but somebody name me this type right here, if you can see. Rough, yeah. So this is going to be the rough ER, and then the smooth ER is going to be this one over here. Rough ER, does anybody know what makes it rough? The ribosomes, good. So the rough ER contains ribosomes that synthesize proteins secreted from the cell. And then the smooth ER is responsible for lipid metabolism and what was it? Like drug detox and breakdown. And then we have our Golgi apparatus here, which on this, right, in the cartoon, it's this one. On the animal cell, it's this one here. So you can see it looks a lot different, which is why you're going to want to be familiar with both of the models. The Golgi apparatus packages, modifies, and secretes proteins and lipids. Then we have our lysosomes. I don't think you guys can see them at all, but they're over here in the cartoon image. These are potent enzymes that can destroy foreign particles or cells. And then the other big thing to know, I don't know if you'll be able to see them in the cartoon, but down here, there's this little black circle with these centrioles on them. To me, they look like little pastas. These are, again, the black one is the centrosome, and then these are the centrioles. These are a like organizing center, and this is what is separated during mitosis. The centrioles are moving to opposite ends of the cell during mitosis. Mhm. And then obviously we have our nucleus, which is the control center of the cell. And then the nucleus is bound by that nuclear envelope. And then also within the nucleus is the chromatin, which is composed of DNA and globular proteins. Globular. And then during cell division, the chromatin network, which are inside of here, coils to reveal chromosomes. So again, that's during cell division, which is mitosis. And then that leads us into mitosis. So the purpose of mitosis, this will be a question on your exam. The very simple, straightforward answer, cell growth and repair. 4 words, full credit. On the exam, again, it's going to ask you, what is the purpose of mitosis, cell growth and repair? And then let's go through the few stages. So a lot of students get confused with like interphase and what it is. This is preparing the cell to go through the process of mitosis. It is not counted in these stages. So the 4 stages are going to be prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes are apparent. So you can see, not really on this image, but if you've come in and taken the extra credit, there's a question and it asks like why is the cell darker during prophase than during interphase? That's because the chromosomes are apparent and there's activity happening. So the party is just beginning to start during prophase. During metaphase, the chromosomes cluster at the middle of the cell here. That's okay. That's usually probably the easiest one to identify is metaphase. And side note, before I forget to say it, on your lab exam, if they ask you to identify one of the stages, it's going to be prophase, metaphase, anaphase, or telophase. There's going to be no early, no late, none of that, just one of those four. And then during anaphase, the chromosomes split into chromatids, and those are then pulled to the opposite sides. Over here are those centrioles I was talking about. Centrioles are like what are being moved during mitosis. Centrosome, I know it is something, but it's like kind of like the holder for them. Like there's not, it's kind of what allows it to move, but it's holding it in place until it's time for mitosis, or I guess until it's time for them to move apart during anaphase.
The cell, let's say, right, if you were to label it there, it would be centrosome.
They would have to be very specific on what they want you to label. If they say like the whole structure, then centrosome, but if they're talking about the specific parts of it, or they may word it like that play a part in mitosis. So label the structure B that plays a part in mitosis. Ols. Good question. Again, so yeah, anaphase, think of it, anaphase apart, and then telophase. This is when cytokinesis begins, as well as the formation of a cleavage furrow, which is right here. And then those chromosomes here start to uncoil. So what a lot of students get telophase and like the completed, what do you call it, a stage of cytokinesis confused. Telophase, they are still technically connected by this cleavage furrow. It is not until cytokinesis that they are separate, and it results in what? Daughter cells, good. 2 daughter cells that are identical to our first cell up here. So cytokinesis has a specific name or division of the what? Cytoplasm, good. So cytokinesis, division of the cytoplasm. That could also be a question. Make sure you are able to identify the stages of mitosis. We do have some of the models here. I want to say there's a specific image they use on the lab, but also make sure you really read the question, because there's a question that will come up on the lab exam, and it's asking you to identify the structures in the cells instead of naming the actual stage that's going on. So you're going to want to make sure you read the question, because they can be asking you to identify like the centrioles here, but they give you this image and you write anaphase. That would be wrong, because they're not asking for the stage, they're asking for the specific structure. And I believe the question or the image that comes up on was on one of your lab quizzes. So you may want to go back and look at those if you have the chance. Okay, and then cell transport. Again, this is copy and pasted from what we did for the lecture review. I did add, let me do here, like a couple questions that may come up from this. So I'm going to run through this because we still need to get to tissues. So cell transport, we have membrane transport processes. And this is, we'll start with passive transport. What is the difference between passive and active? Cellular energy, I think I heard someone say it. So during passive transport processes, there's no cellular energy required. It's in passing, it's doing its own thing. So no cellular energy. Net movement is from an area of higher concentration to lower concentration. And then the next one is diffusion. This is going to be the net movement of substances down the concentration gradient. And so in diffusion, the membrane must be permeable to the substance. So it has to allow that substance in. And then we have another type of diffusion, which is facilitated diffusion, or carrier mediated. It's exactly what it sounds like. It uses a carrier to move across the membrane. So this one has a little bit of help from a carrier protein or a transporter. So that would be the big thing to know between the two, is facilitated diffusion has some help. And then osmosis, what is the difference between osmosis and diffusion? The presence of what? Water, good. So osmosis uses or is the movement of water through a selectively permeable membrane from an area of high water concentration to an area of lower water concentration. And then we move on to active transport processes. These are dependent on ATP or energy use by the cell, since these substances are transported against the concentration gradient. So big thing to know here, active transport needs ATP. So we have two types, or we have a couple types, the primary transport, primary active transport, or the potassium pump. Again, this is an ATP-driven pump, and it requires sodium and potassium, and then ATPase, which is an enzyme. And then the big thing to know here, it pumps 2 potassium ions in and 3 sodium ions out. You see? And then we have secondary active transport. This is when the substances are dragged either in the same direction or the opposite direction as a result of the sodium-potassium pump. And then we have vesicular transport. So we have two types, exocytosis, exiting substances are removed from the cell, and then endocytosis, substances are brought into the cell. And then with endocytosis, we have phagocytosis and pinocytosis, which I know this is a lot of cytosises. Try your best to stay with me here. Phagocytosis is going to be cell eating, and then pinocytosis is going to be cell drinking. One of the other tutors, Joey, he has a really good kind of cheat sheet for cell transport. If you want that, please come see me after or email me and I can share that with you. It's basically everything that's here, just kind of like on one sheet and probably in simpler terms. Okay, and that's it for chapter 3. Again, make sure you're familiar with both images of the cell. This one you'll have to label, this one you'll also have to label. I'm not entirely sure. I would think maybe like one station for each. So about 8 questions total over cells. Okay, now we're going to get into tissues. I like to start with muscle and nervous tissue because to me, those are the simpler ones. They're kind of straightforward. We have 4 specific types of tissues. 2 of them are on the board, so that's muscle and nervous. What are the other 2? Connective and epithelial. So those are the 4 types of tissue. I'm pretty sure that will be a question on the exam. Name the 4 types of tissue: muscle, nervous, connective, epithelium. So muscle tissue, the function is to contract to produce movement. There are only 3 types of muscle tissue. So on the exam, if they're asking you identify this muscle tissue, and you put anything other than skeletal, cardiac, or smooth, the question is wrong, right? Because there's only 3 types. So don't get confused and make sure you really read the question because it may give you clues into what your answer will be. Skeletal tissue is this one over here. You can't really see the picture really nicely, but the posters that are in here and also in the lab are really helpful. These have long cylindrical multinucleated cells. And with the skeletal muscle tissue, it has striations. So it kind of has like little ribs. If you have like a ribbed t-shirt, it kind of looks like that under a microscope. You don't need to worry about these controls just yet, but they are there. And then it's kind of showing you where it's found. So it's found around your bones and within your muscles. So skeletal muscle tissue, where can it be found? Around your bones. I think that's all you need to know from that. Oh, hello. Cardiac muscle tissue is only found in one place. Where do we think that is? Heart, exactly. So if they give you this question and they say name where it's found and you put anything other than the heart, that would be wrong. Cardiac muscle tissue has a distinguishing feature that only cardiac muscle tissue have, and those are these intercalated discs here. They're like little lines, and those are within the cardiac muscle tissue. That will also be a question, and it's either going to be like name the tissue that has intercalated discs, or what distinguishing feature does cardiac muscle tissue have that others don't? The presence of those intercalated discs. Again, you can see it really well on this one over here. These right here are those intercalated discs. And then lastly, we have smooth muscle. Smooth muscle looks exactly how it sounds. It's not striated, so it doesn't have those like ribbed effects that the skeletal muscle tissue has. And it's just very uniform. That one would be this one over here. Very uniform, very nice to look at. Those are found in your organs, around your organs, sorry. So again, muscle tissue, there's only 3 types. So on your exam, if it's asking you to name one of the muscle tissues and it says muscle tissue in the question, you should only be writing skeletal, cardiac, or smooth. Nervous tissue is used to send messages or impulses. You may see it worded like that. This one doesn't usually tend to show up on the first lab exam because we do a whole unit over the central nervous system. But they're made up of neuroglia and neurons, and they look like this over here. If one were to show up on the exam, it would be this middle one here because it has the motor neuron. But where do we think we find nervous tissue? Spinal cord, yeah. Spinal cord, brain, that would be our nervous tissue. And then that's really all you need to know for nervous tissue, is that it sends messages and then where it's found. And that it's one of the 4 types of tissues. Okay, so then we move on to the harder ones. We have epithelial. Epithelial covers the body in organ surfaces and it lines cavities. And it's classified according to 2 conditions, shape and the layers. So shape, we have squamous, cuboidal, and columnar. Squamous, squashed, these are flat. Cuboidal, they look like cubes. And then columnar are just longer in length. With columnar, you can have ciliated and non-ciliated. Ciliated is the presence of the cilia on the top of the tissue, and those are like finger-like projections that allow for transport. And then with epithelial, we have layering. So it can be simple, stratified, pseudo-stratified, or transitional. Simple is going to be that single layer. So you can have simple squamous epithelial tissue. Stratified is going to have 2 or more layers. So you can have stratified cuboidal epithelial tissue. Pseudo-stratified, pseudo means fake. So pseudo appears stratified, but is not. So it looks like it has more than 1 layer, but it still only has 1. And then transitional switches from one to the other. So transitional is usually found in organs or around organs that have a lot of like contraction happening. So Does anybody know where transitional epithelium is usually found? Bladder, good. So our bladder, right, sometimes it gets really, really full and then it'll get really, really empty. So that's where transitional is found. So for all of these, you're going to want to know how to describe it and then where they're found in the body. A good like cheat sheet / hack is on this poster here, and it gives you the answers to all of that. So this one here, take a look at it before your exam or take a picture of it and use that to study at home. And then connective tissue is probably one of the harder ones to learn because it's the most abundant. There's the most of them. And the big thing to know with connective is all connective tissues arise from embryonic tissue called mesenchyme. I know in lab, they did have the mesenchyme tissue actually as out there as a tissue, so you may need to identify it. But the way this will be asked on the exam is like, what type of tissue are all connective tissues arising from? Mesenchyme. You're also going to want to make sure you can spell it. That's a really weird word. Mesenchyme. I've also heard like mesenchyme. There's another pronunciation, but yeah, very weird, but it's there. This is very important to know. Make sure you know this because this will come up on the exam. Okay, and then we have 4 types of connective tissue. We have connective tissue proper, cartilage, and then bone and blood. So I'm going to put something on the screen really quickly. This is like the breakdown of all of the connective tissue types. You're going to want to make sure you know this because this could also be an exam question. Bone and blood are types of connective tissue. They just don't really have anything under it. So you're going to want to make sure you know that. Usually the question that comes up is identifying bone in a slide or in your case pictures, and then what type of tissue. Is this connective tissue? Same thing with blood. So let's see. Connective tissue proper is like the umbrella term, and then it has 2 subcategories. We have loose connective, which is areolar, adipose, and reticular. So again, loose, we have areolar, adipose, which is fat storage, and then reticular. That's loose connective, which just means, right, like all of the cells are kind of loose, floating around. Dense connective, they're tightly packed. This is going to be dense regular, dense irregular, and then elastic. Not to be confused with the elastic cartilage. So you're going to want to make sure you know the difference between the two. And then also over here on these posters, it has like the images of elastic and elastic cartilage. So you're going to want to make sure that you can identify the two. So right, just from connective tissue proper, we have 6 names of tissues. And then on the next slide, we're going to identify a few. But on the exam, a lot of students ask, like, should I be writing areolar loose connective tissue or should I just be writing areolar? That kind of depends on your professor, but I wouldn't write it out unless you're 100% confident, because if you wrote areolar dense connective, that would be completely wrong. But if you just wrote areolar, you could at least get like half credit for it. But if you added dense, then that's just completely wrong. So unless your professor specifically asks you to write out the whole tissue name, then I would only write areolar. Now, this does not go for the epithelium. When we are writing out the epithelial tissue, you need to write pseudostratified squamous epithelial tissue. Okay. So like I said, we have dense, regular. This is found in tendons, ligaments, which makes sense that it's really dense because we want our ligaments to be stretchy, but also like really strong. And then irregular are found in the joint capsules, which we get into in the next unit, but you do need to know irregular dense tissue. And then the elastic tissue, where these are found. Yeah. Okay. So then those would be all of the proper connective tissues. We then get into the cartilage, which we also have 3 types. We have hyaline cartilage, elastic cartilage, and then fibrocartilage. Hyaline cartilage, I'm going to point at George up here, the skeleton, are these right here. So you can see that your ribs do not directly attach to your sternum. They attach via cartilage. This is called costal cartilage or hyaline cartilage. So on the exam, it'll ask you where is hyaline cartilage found? Ribs or at the end of long bones, in your nose, and I believe your trachea as well. But you would only need to name one of those, so whatever is easiest for you to remember. It might be nose, just remember nose. And then we have elastic cartilage. This one is found in your ear, and that's the way I always remember elastic cartilage, ear, so the E's. And then the fibrocartilage work as cushions. I don't think we have a. So between each of your vertebrae, so your spine, there's 24 individual bones. There's a piece of fibrocartilage in between each. These are called the intervertebral discs. Those are made up of the fibrocartilage, and that's what allows you to like jump up and down and not have all of your bones like clanking on each other. It has that cushion there. And then lastly, connective tissues, bone, and blood. Those are pretty easy to identify. The bone is this one here, kind of looks like a tree. If you cut open a tree, blood, you can see all of the characteristics of the blood cells in here. So bone and blood are connective tissue types. I have a few more minutes, so we're going to try a few, if you can see that far. Number 1, can anyone describe what this one is or name it? Take your best shot. Hyaline, good. Hyaline cartilage, this is number 1. You can tell by the like where the nuclei are and then kind of just the anatomical uptake of it. It's just one of those that when you see it, you should be able to identify it. Number 2, bone, good. This is bone tissue. What type of tissue is this? Connective, good. Number 3, adipose, good. Where is adipose found? Yeah, breasts, fat, good. Number 4 is up here, blood. What type of tissue? Connective, good. Oh, I almost did it. What about this one right here? I think I hear it. Dense, regular, good. Dense, regular, what? Connective tissue, good. And then this one down here, dense, irregular, connective tissue, good. And then you can see the difference between the two. This one is just very put together, very uniform. Dense, it's very tightly packed. Regular, irregular, there's no uniformity, it's just all over the place. And then 7, areolar, good. This one is going to be loose areolar connective tissue. Very good. This is what they should kind of look like on your exam. I do believe they're using pictures this semester, so this is what you should be looking for. And then some final tips and takeaways. Make sure you show up to your exam on time because you only have a couple minutes per station. You do not want to miss any because you were late. I do believe it's up to the professor whether or not they let you like go back around and give you time to go back around and fill out any that you may have missed. But you do want to make sure you're on time. And then try out the practice exam available to you here in the Science Learning Center. It's a good indicator of how much more you need to study or what material you are comfortable with. We do also have the microscopes and we have most of the slides that you'll be seeing on the exam. So yes, can I borrow that real quick? If you have not picked this up from the library, make sure you go and get one. It's available to you as anatomy students. Specifically, it's really helpful with the tissues. Oh gosh, because the images are very like HD and very helpful. And this will also become especially helpful when we get to the bone section, so the next unit, because you need to know like specific areas, but we won't talk about that right now. But yeah, So I know my class is taking their exam in like 30 minutes, but in the meantime, if anybody has any questions or you just want to hang out in here, I will be in here. What? Not for the lab exam. Or at least, let me, see,